In this video, we're going to go over the difference between medicine and drugs, and we're also going to take a closer look at the drug discovery process and the steps that are involved in order to get a medication to market. So let's start with our definitions of medicine versus drugs. So a drug is a chemical that's going to affect how the body works. And that includes changes for the better, but it also includes changes for the worse. So it doesn't necessarily have a beneficial effect. Whereas a medicine is something that will improve health. So it's always a beneficial effect. It can be natural or it can be synthetic, and we'll look at various examples of both. Can also contain non active ingredients. And that beneficial effect is what's called the therapeutic effect. So we're going to take a look at this a little bit closer as we explore different medications. Now, in terms of how pharmaceutical products work on the human body, essentially most of them, I would say a good 70% or so of them at least, are targeting membrane proteins within the cell. And so the drug is going to produce an effect on the body by interacting with a particular target molecule. This is usually an enzyme or a receptor. So receptors are typically membrane proteins. They're embedded within the cell membrane. And so when the drug comes and binds, it can bind to either the active site of the enzyme, or say it's binding, this is a receptor protein and it's binding at this site here, then it can produce its effect. And it'll do it in two ways. So when it binds, it's going to either stop it from functioning. So if we want the function of that particular enzyme or receptor to slow down, we'll get it to stop functioning. Or it could do the opposite and stimulate it to produce a larger biological effect. So it kind of depends on which way we're going, what the particular effect is here. Now, in terms of how medications or pharmaceutical products get to market, there's four sort of main stages of drug development. They're discovery research, development research, regulatory review, and then ongoing is post-marketing monitoring. What is really, really shocking is on average, it takes 10 to 12 years. So a really, really long time to develop a market from its to develop a drug, from its first identification to when it appears on market, when it's available for patients to be able to use. So we're just going to take a look at each of these stages in a little bit more detail. The first stage is discovery research. This is where we identify lead compounds, not lead, but lead compounds. And so these are compounds that are being shown to have biological activity. And usually they're derived from plants because plants have lots of really special properties that affect biology. And then once those compounds are being identified, they will be optimized. And so basically, we'll take the compound and we'll create many chemically related compounds. They're called analogs, and they're made and tested and usually done through a couple of techniques. It's called combinatorial chemistry. So they're just combining different, say, functional groups on these different compounds, and then they're doing some high throughput screening. So this is usually done with various different robotics, and it's all sort of very, very automated. And then we'll do initial testing of the potential medicine. So when we've identified ones that have got good promise, we'll do testing on animals. This is done under really, really strict legislative control and helps us to determine the dose that should be administered in human trials. So once we've gone through all that process and done animal testing, it's time to move on to stage 2, which is development research. And essentially, this is 3 phases of clinical trials on humans. Each phase is going to increase the number of patients tested. And really what they're looking for is 2 things here. They're looking for safety, and safety is first. And then they're also looking for efficacy. So, how effective is the potential drug? So, phase one will start with a very small sort of population, 50 to 100 healthy volunteers. And that's because we're looking at safety only in phase one. If it's determined to be safe or safe enough to move on to phase two, then our study gets a bit bigger, 200 to 400, and now it's patients. So these are ones that are sick, have the disease or illness that would benefit from the potential medication that has been discovered. So they're still focused on safety here because we're keeping it relatively small, but this is our first sort of glance on seeing how what the efficacy is like of the particular drug. If we get past that, we move on to stage 3. This is a very large study, over 3,000 patients, and it's a double-blind study. So what that means is that half of the population is given the drug, the other half is given a placebo, and it's double-blind because neither the doctor nor the patient knows what has been given. Now, I did mention placebos in there, so we do have to consider the placebo effect when we're doing these studies. And the placebo effect occurs when a person experiences a positive therapeutic effect because they believe they've been given a drug, although the substance given in reality is not a medication, but it's really the placebo that's being administered. So essentially, it's the body fooling itself into healing. This is really, really prevalent with pain and pain medications. So you do have to consider this when you're carrying out clinical trials and analyzing the results from them, because about a third of the control group taking a placebo is going to show some improvements. And that's just a fact that's used in all sort of major clinical trials. So stages 3 and 4 kind of go hand in hand. This is the regulatory review and the post-marketing monitoring. So the regulatory review is once you've done all the studies and everything looks good, this is for you to apply to be able to market the product. And then stage 4 post-marketing monitoring is collection of any adverse drug reaction data. So it's really important because we've done a large study, but it's still not that big as compared to our human population. So we do need to continuously monitor drugs as they are released out into the market to make sure that they're not causing any severe kind of side effects that weren't caught in those clinical trials. So a really good example of that is thalidomide. And we'll hear a bit more about thalidomide as we sort of talk about medications and drugs, but this was a morning sickness drug that was prescribed to pregnant women. And really kind of an interesting case because it got through all of the clinical trials and looked like it was safe and efficient, but it started causing severe birth defects. There were things like missing or malformed limbs. Many babies didn't even survive infancy, and that's because there's actually two different isomers, stereoisomers of thalidomide. So one of them is safe and one of them is not. And so if you had a racemic mixture of this drug and you gave it to a pregnant woman, it did cause these defects because of the one particular stereoisomer of this drug. So really, really important that we're continuously monitoring and making sure that the drugs that are going to market continue to be safe and efficient for patients. So that's it then for this video. We've looked at the difference of medicine and drugs as well as the drug discovery process. We'll see you in the next one.